Hi, my name is Heidi Rehm, and it's a pleasure to give this educational session on using genomic resources to interpret the clinical significance of rare variants. So I'm going to talk mainly about 4 different resources that I think are critical in variant classification. That includes ClinGen, ClinVar, the GenCC, and Nomad, and I'll tell you more about these resources throughout the talk. First, I'll start with ClinGen. Really, the goal of ClinGen is to create an authoritative central resource that defines the clinical relevance of genes and variants for use in medicine and research. And this program started in 2013. It's been primarily funded by the NIH through 3 core U41 grants, but more recently, a number of disease-focused grants have also been awarded, initially 3, and now there's another round just coming out this summer. And over on the left side here, you can see a lot of the different areas of work that we do. We aggregate genomic and health data from different sources, patients, clinicians, labs, and researchers. We curate that data based on ClinGen's critical questions, where we're curating for gene disease validity, variant pathogenicity, dosage, sensitivity of particular genes, and clinical actionability. Is this information useful for a patient? And then we disseminate that expert knowledge to the community with the goal of improving patient care. All of the information that we curate is on our clinicalgenome.org website, and all variants are also submitted to ClinVar. In terms of a lot of the work is done through the formation of expert panels in different disease areas, and what you can see on this slide, that's a picture of our website, is that we have 13 different large clinical domain working groups, and then those clinical domain working groups further develop more focused expert panels in either gene curation or variant curation, or sometimes doing both. And so you can see we have 77 different expert panels across these different areas. To date, those efforts involve over 1,700 investigators from 44 countries around the world. So really global participation, and it's been great to see the community involved in this effort. Now, as I mentioned, one of the areas we do curation in is gene disease validity. So is this gene that's been reported as causal for a particular disease, is there sufficient evidence for that claim? And we published a semi-quantitative framework to really look at this evidence and classify it, and we score genetic evidence, so case observations, segregation in families, case control studies, things like that, as well as experimental evidence, expression of the gene, model organisms, rescue studies, those sorts of things. And once we score all of that evidence, it then leads to a category, one of the ones shown here that I'm circling, either definitive, strong, moderate, limited, only an animal model but no human evidence, no known disease relationship, disputed, or refuted. And what you can see here is of the 1,320 gene disease pairs that we've evaluated across these many expert panels, that we've scored in these relative categories. And so one thing that's important to note is about 1/3 of them, or 30% of these gene disease pairs, have landed in the insufficient evidence area. So it's really worth pointing out that this curation is really important, because not every gene that's published as a disease gene per se actually has sufficient evidence for that claim. And of course, all this information is then put on our website. I'm showing a screenshot from the stats portion of our website, where we show curations from each of the different expert panels doing gene curation. I couldn't fit them all on the slide, but this is a sampling. And what you can see is that for each of the disease areas, they all suffer from some degree of lack of evidence for certain claims. I think the worst is Brugada syndrome, where all but one of those genes were actually found to have insufficient evidence and were disputed by the expert panel. Others are much better. Hemostasis thrombosis group, is much more definitive set of genes, but it varies across all areas. So how are labs actually using ClinGen's gene curation results? Well, here's an example from Invitae's website. I mentioned the Brugada syndrome curation effort, and right after the experts released their results showing that only one of those genes was validly implicated in disease, Invitae changed its primary panel. Only that one gene is now included as the default when a physician orders a test for a patient. However, they do have the option to add on preliminary evidence genes if they feel that they want to get results back on those genes. So it's a balance in terms of thinking about whether these genes could ever be future genes. Some that are limited may move on, although I can tell you in our experience, most genes that fall in limited or below never actually move up unless they're very recent publications. As I mentioned, you can go on our ClinGen website and all of the gene curations are there. You can go onto the website and type in your favorite gene. Here I've just shown an example of typing in MYBPC3, a gene for cardiomyopathy. And I do want to point out here in the gene disease validity curations that you can actually have more than one gene disease claim for a given gene. And in this case, they can be different in terms of the level of evidence. So you can see here that the experts viewed the association to hypertrophic cardiomyopathy as definitive, but the association of arrhythmogenic right ventricular cardiomyopathy as limited, and likewise the relationship to dilated cardiomyopathy. So those relationships can vary across different diseases. You'll also note that this gene has also been curated for dosage sensitivity, so a full deletion of the gene, as well as triplosensitivity, a full gain of a copy of the gene. And that was found to have evidence for haploinsufficiency, but not triplosensitivity. And there is also a curation for actionability that is pending. ClinGen is not the only group that curates gene disease relationships. In the last couple of years, we formed an organization called the Gene Curation Coalition that really brought, at the beginning, we brought all of the public gene resources together, and with the goal of clarifying the overlap between those efforts, understanding how they classify genes, and then we developed consistent terminology so that we could interoperate between the different gene level resources. And now we've been focused on posting gene curations from any group willing to share their data. And we've added more recently many clinical labs that, although they hadn't maintained public resources with their data, they were willing to share it. And so this has been a work in progress. We've launched our GenCC database now, and we are now working to collaborate on gene curation projects between the groups. So I mentioned we had to harmonize the terminology. What you can see here on the left is that many of these different groups were using different approaches for how they documented the evidence for the gene disease relationship. Some were terms, the ones I showed you for ClinGen. Others were using colors, but in some cases those colors were the opposite in terms of what was strong and what was limited. So you can imagine the confusion for the community trying to navigate across these different resources. So by bringing all of these groups together, we could work towards harmonizing the terminology. We actually did a Delphi survey and did 3 rounds of that survey, first within our community, through our extended membership, and then with the international genetics community with lots of input. And through that effort, we arrived at the harmonized set. It's very similar to the ClinGen set, although we did add the animal model only category and differentiated that with no known human disease relationship. This is just a screenshot of our GenCC database, which is now open for browsing. We like to call it the ClinVar for genes. So, the different groups that submit are submitting their claim for gene disease validity, similar to a group submitting to ClinVar for their claim for variant pathogenicity. So this is a screenshot, and I've expanded for GJB3, where you can see both summaries of each of these genes, and then you can expand and see what claims each of the groups that submitted have made, and sometimes there may be, as I mentioned before, more than one claim for a different condition. In this case, erythrokeratodermia variabilis, or non-syndromic genetic deafness. This is just a summary of all of the submissions to date. So we have over 7,700 classifications that have been submitted. You can see the breakdown across the different groups that have so far submitted. We're still gathering more data from each of these laboratories and organizations that are submitting. And the next step will be to start to resolve conflicts between our gene disease validity claims from different groups. And this is where we're really working together as a community. Next, I want to move on to our efforts in variant pathogenicity assessment. In this case, we take a more multi-pronged approach. There are just too many variants in our human genomes in order for experts to curate all of them. And so therefore, we have to take a multi-pronged approach, both in terms of using common standards, terms for variant classification, as well as rules. And we've done this initially through the publication of a joint consensus standard from American College of Medical Genetics and Genomics and the Association for Molecular Pathology, where we defined 5 terms for pathogenicity, as well as different types of evidence and its strength, and how to combine that to arrive at these different categories. And this framework has largely been adopted worldwide, but there's still subjectivity to the application of these rules. And so we've had, through ClinGen, a sequence variant interpretation working group co-chaired by Les Biesecker and Stephen Harrison, who have been working to refine and evolve these guidelines as they've been tested and deployed by the community, as well as review and harmonize gene and disease specifications that are provided by ClinGen's variant curation expert panels for each of their genes or diseases. And then we're gradually moving the framework towards a more quantitative framework using a Bayesian framework. All of the different general rules as well as disease-specific specifications are all on the ClinGen website. I've just pasted a few papers here, but there are many there on the website for you to be able to access. In addition to common standards, we also work to get the community to share variant classifications from each of their labs, and this creates both transparency. We can look and see when we say the same thing or something different. It also crowdsources the work. If you've already gone through the literature and brought the evidence together, then when I see a variant, I can simply add my evidence to the evidence you've already gathered, as well as check and confirm that you've gathered it from all the sources. So it allows us to really work as a team across different laboratories. And to date, as of just last week, there's now over 1.5 million submissions on nearly 1 million unique variants in ClinVar from submitters from around the world. Most of this data comes from clinical testing, although research groups and other online resources, as well as our expert panels, also submit to ClinVar. Interestingly, nearly 84% of the data in ClinVar is actually from the top 20 submitters, 15 of which are commercial clinical labs. And I think it really speaks to the goodwill in our global commercial lab testing community that so many of these labs are voluntarily sharing their data with ClinVar to enable a more accurate and robust knowledge base for patient care. My guess is many of you have used ClinVar, but if you haven't, you can go to ClinVar and you can type in a variant that you're searching for. I've, in this case, typed in the M34T variant in the connexin 26 gene. And I do want to point out that ClinVar, although ClinVar doesn't make its own interpretations or classifications of variants, it does aggregate and calculate an overall classification. So if all the labs agree with each other, it will give that single overall classification. Or if there's an expert panel that's submitted to ClinVar, that expert panel interpretation overrides all of the others. And this variant is an example where there are actually 36 submissions from different labs. They don't all agree with each other, but we gathered experts from the community together, and our hearing loss expert panel evaluated all of the evidence from many labs and were able to come to an overall conclusion of pathogenic for this variant. That said, you can still scroll down and see the different interpretations. Also, if you look at the different nomenclature, so many genes have multiple transcripts. And one of the ways that we're getting the community to harmonize around a single nomenclature for individual variants is to use what we call the main select transcript. That is a default reporting transcript that's from the MAIN project, which stands for Matched Annotation from NCBI and EBI. And there, these two groups that curate transcript information have come together, ensured that those transcripts match 100% from end to end, and also have biological evidence as really the key biological transcript. It doesn't mean there aren't other transcripts that are also important for that gene and may contain exons that are uniquely present in other transcripts, but this has been chosen as the default primary reporting transcript when a variant is present in all of the transcripts. So we encourage you to use the main select set. Now I should point out main select is mapped to the genome build 38. So if you're not yet on build 38, some of the transcripts don't have a version on build 38. So I encourage you to move to build 38 if you're still on 37. Now the next way that we work on generating accurate variant classifications is actually to facilitate interlaboratory conflict resolution. So if you look across ClinVar, what you see is, well, one interesting fact is that 75% of the submissions to ClinVar have only one submitter, which emphasizes just how rare most genetic variation is, and that we don't have the option to see it twice. However, about 25% of variants do have more than one submitter and are in this part of the pie. And while a fraction of that is concordant, so we say about the same thing or roughly the same thing, about 20%, for about 5% we're not concordant, even across the major sort of categories. And for 1%, there are medically significant conflicts. So one lab is saying the variant is pathogenic or likely pathogenic, and another lab is saying it's either uncertain, likely benign, or benign. Those are what we call medically significant conflicts. Right now, or as of last week, there were over 12,000 of those conflicts. Or if you exclude the 0-star submitters that don't have assertion criteria or provide evidence, then it's about 7,500 just looking at those submissions. So it's a lot of work, and it highlights the fact that we do need to work together as a community to resolve these differences in variant interpretation. And through ClinGen, this is work led by Stephen Harrison and Jill Dolinsky. We've been going through various phases of discrepancy resolution work. The first round we did with just 4 labs and with a really deep dive to look at whether we could come to concordance. And we were able to come to concordance on 87% of variants if we both groups, all 4 groups, re-looked at all of the variants that were discrepant. However, it was a lot of work when you had to reevaluate every variant. In phases 2 and 3, we moved to an outlier approach where the lab that was different than the rest of the labs first evaluated the variant, or sometimes if a lab had an older classification compared to others, they would first evaluate it. And we did that with a set of 41 labs and now a larger set of 108 labs. And through that effort, although it doesn't resolve quite as many as if everyone reevaluates, it still resolves quite a large percentage of these. I should also point out that even when we don't organize labs coming together, just by virtue of being able to see evidence across laboratories and what was curated, that over time resolves about 24% of discrepancies. So really underscoring the importance of global sharing of variant classification. And then lastly, and most critically, this is where we engage experts. As I mentioned in that couple of slides before, for the interlab conflict resolution, we don't always come to 100% agreement. We did in the All of Us program, partly because we had to, but otherwise across labs, we don't always agree. And this is where bringing experts in and coming to consensus is important. And what these expert panels do within ClinGen is they first define gene or disease-specific specifications of the ACMG AMP guidelines. So what are the allele frequency thresholds for calling a variant benign for that gene or that disease? Which are the hotspots for the gene? What functional studies are valid for use and at what strength of evidence? So these are all the things that each of these expert panels specify. And they go through a series of steps of approval. First, defining their work groups and their scope of work. The conflicts of interest are transparently presented. They develop their rules for variant classification. They pilot those rules, get them approved by our sequence variant interpretation working group, and then they can begin implementing. And that allows this rigorous process of rule development allows them to be classified as expert panels in ClinVar and also recognized by the FDA in terms of their FDA recognized database status. So to date, there are over 12,000 expert classified variants in ClinVar. The goal is not to classify every variant, as I mentioned earlier. There's a lot of work that goes into these variant classifications, especially when they're differing between laboratories. So we really target those most important variants that we need to come to resolution on. You can get the detailed evidence, although they all go into ClinVar. There's a link from ClinVar back out to our evidence repo where you can get a lot more detail about the rationale for individual rules being applied and all of the evidence and publications, etc. Now, we don't just focus on discrepancies. Sometimes there are variants that everybody agrees are uncertain. But by bringing all those groups together and their evidence together, we can move variants that maybe everyone says are uncertain, but move them up to likely pathogenic or down to likely benign or benign. And this is just a graph from one of the publications from one of the expert panels showing a number of variants that were able to move to likely pathogenic by bringing data from different laboratories together. So with that, I want to acknowledge the many individuals that are part of ClinGen. This is our steering committee with multiple PIs from the different grants that fund ClinGen. But we do have lots of working groups that develop the approaches that we use across these different areas of curation. And of course, as I mentioned earlier, we have over 1,700 people that are participating in our expert panels, all contributing to these efforts as a global community. Now, to give you a sense of how we use a lot of these resources I've been talking about and some more that I'll show you, I'm going to go through one of our cases from our Rare Genomes project where we enroll patients who have not been able to obtain a diagnosis through other clinical testing. So in this case, the proband is shown here, a 2-year-old male who had a congenital presentation of profound hypotonia, macrocephaly, frontal bossing, nystagmus, retracted testicles, and delayed bone growth. He had had prior genetic testing that was all negative. And we, after sequencing he and his parents as a trio, we did a typical search looking for dominant and recessive causes of disease and identified one de novo variant. Now, most every exome or genome we sequence, you find on average one de novo variant. So although somewhat suspicious, it's not a guarantee. But that de novo variant was in the GNB1 gene. At the time that we identified this, that variant had been implicated in disease, although the variant itself was not known and was not submitted to ClinVar. But it was absent from Nomad and was predicted to be damaging. If you look at Nomad, we now have all of the ClinVar variation track in Nomad, so you can look and see, in this case, the variant was located here. It actually now is in ClinVar. At the time it wasn't, but you can sometimes help think about the significance of a variant by its location. Is it near other pathogenic variation like is shown here? It seems to be a hot spot. It's also in an exon that's well expressed in the gene, ensuring that it's relevant to the function of the protein. You can also see that this gene is highly constrained, particularly for loss of function variation, but even constrained for missense variants. If you look up the gene in OMIM, it is associated with mental retardation, and if you look at the clinical synopsis in OMIM, you'll see there is overlap between some of the clinical features and our patient. That said, hypotonia, nystagmus, postnatal growth, these aren't highly specific phenotypes, so you might want to dig a little bit more. Going into GenCC, there's gene disease validity curation, and in this case, if you look up GNB1, Ambry has submitted a classification for this gene and evaluated it in 2018 as moderate evidence. And I should note that not all of the OMIM submissions are in GenCC yet, so it's useful to go both to GenCC and OMIM if you're looking up a gene and you don't find it in GenCC. Now, one resource that I do like to go to, particularly to get better case level data and phenotypes, is the Decipher database from Helen Firth and Matt Hurls in the UK, and they have a lot of particularly neurodevelopmental cases that are well phenotyped that are in that database. So here I'm showing you searching for GNB1 in the Decipher database, and there's a track here above the gene that shows all of the Decipher cases. You can dive into those cases and see aggregate statistics as well as individual level detail about each of the cases. You can see that 90% of those cases were de novo, 10% inherited. You can see individual phenotypic features that were seen in patients with variants in the GNB1 gene. And you can get a list of individual features and how many patients with a mutation in that gene had those features, as well as the percentage of cases, and then directly compare that to the overall set of 27,000 phenotype patients in Decipher, and they will give you a p-value to show whether that is or is not correlated with disease. And you'll see that if we dive a little deeper into the specific phenotypic features seen in the case that I mentioned, and those are listed here correlating with, this is the Rare Genomes Project patient. These are the same features, and they have been seen in at least 1 to 2 other GNB1 patients. You'll see the percentage of patients compared to the overall percentage in Decipher, so it is higher. There are statistically significant p-values for these particular phenotypic features, so it gives you more confidence that this may be the causal variant when you're seeing the correlation in phenotype. Now, occasionally you'll find a de novo variant in a constrained gene and it's looking very suspicious, but you go into all these databases and it's not, and there's no gene disease association. In those cases, you'd want to enter that gene into the matchmaker exchange, and that allows you to find other cases where they may have flagged a candidate gene that overlaps with your gene list, 1 or more candidates you might have found, and then you can, by being matched with other cases around the world, compare the phenotypic features to look for overlap. So this database has over 10,000 candidate genes entered it from thousands of patients, from thousands of contributors from all around the world. And if you aren't already affiliated with one of these nodes that participate in the Matchmaker Exchange, it's easiest to register in Gene Matcher, and you can just enter your gene, and then you will get matched with any other cases for which a candidate gene that matches your gene has been flagged in one of these other databases. And then you'll get sent an email along with the other person who submitted that gene to the database. So a really great way to build evidence for gene disease causality. And with that, I just want to finish talking about ways to get involved, to share your knowledge. It takes a whole community to advance our understanding of the genetic causes of disease. So if you're interested in getting involved in ClinGen, you can go to our website to the Start button, and you'll find ways to get involved, such as volunteering as a biocurator or joining an expert panel if you're an expert in a particular disease area. And you can find the list of expert panels on our website. You can also submit variants to ClinVar if you classify variants in your laboratory. You can join the GenCC if you classify genes. And if you're a clinician, you can refer your patient to a patient registry that collects genetic results and shares them with ClinVar. If you're not aware of a registry for the particular disease, you can send your patient to the ClinGen registry called Genome Connect, and we will share anyone's genetic results with ClinVar. We also partner with disease-specific organizations to help them share patient data. And with that, I would just like to acknowledge many of the projects that I've talked about today: ClinGen, the ClinVar database at NCBI, the GenCC, our Nomad database, the Rare Genomes Project in affiliation with our Center for Mendelian Genomics, the Matchmaker Exchange, and of course funding from NHGRI, the Chan Zuckerberg Initiative, and the Wellcome Trust. And thank you very much, and I look forward to taking any questions.